Okay. The pathways for the resistance, وin particular resistance against the PCR-APL ممكن تكون, let's say, dependent on the PCR-APL kinase or independent. Okay, dependent, وبرضه كمان هاي approaches can be seen for the emergence of resistance against other let's say inhibitors, سواء كانت kinase inhibitors أو any other inhibitors of other enzymes, واللي هي amplification of the enzyme itself أو overproduction of the enzyme, اللي هي حتى لو كان some of it resist أو some of it would be inhibited, there is still more to aid in proliferation of cancerous cells. بحيث إنه هاي therapeutic dose تصير ineffective anymore because we would need, let's say, a much higher dose to inhibit all of the produced faulty kinases, okay, أو overactive kinases. Another approach for the resistance or another mechanism for resistance اللي هي single mutations أو mutations in the structure of the kinase أو حتى target protein in general, such mutations would lead to lowering or diminishing the binding of the inhibitor to the kinase. هلا بنحكي عن that specific mechanism regarding الإيماتينib. أحيانا الcancerous cells ممكن تصير تعتمد على other pathways such as in the case of the PCR-APL بتصير تعتمد على other signaling pathways or other kinases that will lead to the same biological effect, okay, such as in the case of the PCR-APL cancerous or those, let's say, cancerous cells where we have the PCR-APL kinase, they would, let's say, then relate على ال SRC kinases, which also are important for proliferation of cancerous cells. Okay? هلا ال independent mechanisms or those mechanisms of resistance that has nothing to do with the enzyme, specifically or in not specific mechanisms. So you can see them also in almost all kinases or in almost all cancerous cells. هلا for imatinib in particular, for this drug to enter cancerous cells, it is actively transported via, let's say, OCT1 protein. Okay? هلا cancerous cells may mutate those proteins so that the entrance or the binding affinity of those proteins to imatinib or the drug would be less, ومعناها less of the drug will be transported inside the cells. Or, as you can see, or as you all know, there are efflux proteins or proteins that aid or that bind to drugs after they enter the cancerous cells and they transport them outside of the cell. يعني they will not give the drug enough time inside the cell to interact with its target. Okay? هلا ممكن يصير في عندي mutation أو ممكن يصير overexpression لهاي الefflux pumps، ممكن يصير في increase أو mutations بحيث إنها تزيد من binding of the drug to those efflux proteins، وبالتالي بيزيدوا من efflux of the drug to the outside of the cell. هلا one mechanism اللي احنا interested فيها regarding الإيماتينب mutation اللي بيصير for the APL kinase, one single mutation which is the main reason for resistance against الإيماتينب، واللي هي change of the threonine, the gatekeeper residue, into isoleucine. هلا when you see T315 mutant معناها إنه this kinase has changed its threonine that has a sequence number of 315 to isoleucine. Okay? What is the difference between the side chains of those two amino acids? Yep. Will isoleucine, it's a single, let's say, carbon chain. Okay? So the variations is that threonine has a hydroxyl group that is able to form hydrogen bond interaction. When you change it to isoleucine, isoleucine does not have any functional groups except carbons and hydrogens, which are not able to form any kind of hydrogen bond interaction. معناها in this mutant enzyme or kinase, when the threonine is replaced with isoleucine, that hydrogen bond interaction, which is crucial for the binding of imatinib to APL will be lost. And when this interaction is lost, the binding affinity of imatinib بتقل بشكل كبير جدا. That is why then the cancerous cells become resistant or imatinib becomes ineffective against those cancerous cells. Let me show you the crystal structure بس عشان أذكركم بهاي gatekeeper residue. And the interaction it forms. So that's the APL kinase bound to imatinib. Let's say, close into the drug. And as you can see, it's a type 2. That's the ring in here, the pyrimidine that binds to the hinge region. This methylphenyl reaches all the way to the hydrophobic pocket. And the amide phenyl piperazine rings can reach to the allosteric, let's say, pocket inside the APL enzyme. So it's a type 2 inhibitor. Okay? هلا remember حكينا على that's the hinge. This loop in here behind this pyrimidine ring, that's the hinge. That's the loop that connects the N loop to the C loop. Okay? هلا على امتداد الhinge region بيكون في عندي let's say a residue at the opening of the hydrophobic pocket و that specific amino acid احنا بنسميها gatekeeper residue. It's this one in here. خليني اعمل كليك عليها على اساس نعمل زوم ان نشوف الانتراكشنز اللي بتعملها. Okay, so that's the threonine in here. And as you can see, the threonine is forming a very important hydrogen bond interaction with this nitrogen that is connected or attached to the pyrimidine ring. This is a very crucial interaction. If you lose this interaction for imatinib in particular, الافينيتي عندي بتقل بشكل كبير. Okay? That is why if you change the threonine into isoleucine, which is hydrophobic in nature, unable to form hydrogen bond interaction, معناها ما رح تعمل interaction مع هاي النيتروجين, that crucial interaction will no longer exist, will binding affinity of imatinib to that mutated APL will be way much less. Okay? طيب. Yes. هلا الهيدروفوبيك باكيت او سوري هلا الهنج region is this one in here, that one. Okay, this is at the front of the يعني هي when they flip, let's say the structure تختلف. هلا it's the area that is in front of the, let's say, adenine pocket. والامينو acids في ال APL kinase اللي موجودين في هاي الباكيت او in that, let's say, space are hydrophobic in nature. طيب. Now, if you are in those who developed imatinib place, what would you do to resolve this issue? شو ممكن تعملوا؟ بإيش ممكن تفكروا؟ That's, let's say, a good approach. ممكن وارد. Okay, مع إنه الهيدروفوبيك interactions in terms of way much less versus أو مقارنة بال let's say الهيدروجين bond interaction. يعني البوينت is اللي هي to modify the structure of imatinib بحيث إنه يا إما إنه to overcome that interaction بحيث إنه حتى يرتبط بالwild type وبالmutant type of APL، أو إنه أغير الستركشر بحيث إنه أزيد الoverall affinity اللي حتى لو خسرت الانتراكشن مع الثريونين still يكون في عندي other interactions ممكن تعوض. Okay, so this is what they've done. They took the imatinib structure and they've started, let's say, modifying or optimizing the imatinib structure. This gave rise to the development of two derivatives of imatinib اللي هم nilotinib والdasatinib. Look at nilotinib structure and compare it with imatinib. شو الchanges اللي عملوها؟ Yep. So that's the piperazine in imatinib. They've changed it to imidazole. What else? مثل imidazole. They've added a trifluoro, let's say, methyl على الphenyl ring. Okay? هلا this is based على إنه if you would look even more closer to the خلينا نحكي active site أو to the hinge region أو منطقة الهنج بال APL kinase, there is a pocket at where they've added the trifluoromethyl that is hydrophobic in nature. يعني بنفس الالوستريك let's say site there is a hydrophobic pocket اللي بتتفرع من الالوستريك site, and when they've added that trifluoromethyl it was able to reach that hydrophobic pocket, increasing the overall binding affinity of the drug. In nilotinib versus imatinib has a binding affinity that is almost 20 to 30 folds أكثر. Okay? طيب النيلوتينب type 1 or type 2? Yeah, it is still type 2. Okay? هلا look at the interactions it forms. Do you think it is active or inactive against the mutant APL أو T315I mutant? طيب what about البايندينج affinity رح تكون أقل ولا أكثر ما بين مثلا الوايلد تايب والميوتنت تايب؟ بتكون much less. هلا if you look at the interactions that are shown in here where nilotinib forms with the APL, it still forms hydrogen bond interaction with the threonine 315. Okay, so for those mutant APL kinases where the threonine was replaced with isoleucine, this compound البايندينج affinity بتكون كثير أقل. Okay, طيب الداساتينب. Look at dasatinib and tell me what changes have they made. Does the structure even look now any close to imatinib structure? Yeah, they've made a lot of changes. طيب, does it still, based على what is shown in here, does it still form an interaction with the threonine 315? Yes. So the binding affinity of this drug to the mutant and the wild type APL, are they the same? No. Okay. Still, it's almost inactive towards those, let's say, mutant or mutated APL kinases. لكن overall البايندينج affinity بشكل كثير أكبر أو هي أفضل حتى من النيلوتينب وفور شور من الإيماتينب. Something else, if you recall, واحدة من الميكانيزمز for resistance against الإيماتينب إنه أحياناً الكانسر cells بتصير تعتمد for its proliferation على ال SRC, let's say, kinase. هلا the good thing about dasatinib when they developed it is that it binds to both APL and SRC. Okay, so that is why the effectiveness of dasatinib is way much more than imatinib and nilotinib because it has a dual specificity على two kinases that are known to be implicated in leukemia. Okay, طيب هلا مكتوب عندكم بالسلايدز انه it binds to both active and inactive forms of the APL. انتوا شو رايكم؟ طبعا هاي المعلومه بالليترشر القديم صحيحه او مش صحيحه، يعني الاكسبيرمنتس have shown that it can bind to both، لكن المور thorough accurate studies shows لا، انه it favors one conformation versus the other. برايكم what conformation it favors؟ ال inactive conformation. طيب حددوا وين الرينج اللي بترتبط بالادينين بايندينج باكيت. What about this one? Yeah, it is. There's a nitrogen that is part of the, let's say, an aromatic system. So this one is actually where adenine binds in. أو أسفة, where this ring system is what binds into the adenine binding pocket. فبالتالي whatever is attached to it can reach only to the hydrophobic pocket. لو في an extra ring in here, it may extend to the allosteric site. Okay? لا, هي الكريستال structure أو الكريستال structures that has this drug co-crystallized with the APL kinase shows that it's a type 1 inhibitor. The one that forms interactions مع adenine binding pocket, actually it's this one rather than this one. And this one reaches the hydrophobic pocket. But what about this part of the drug? Yes, that protrudes outside of the ATP binding site into the solvent region. Do we have any ionizable group or groups? Which amine? Peprazine. Peprazine has 2 nitrogens. A nitrogen. Yes, this one, the aliphatic nitrogen. This one is an aromatic nitrogen or an aniline-like nitrogen where the lone pair of electrons are withdrawn inductively and via resonance. Okay. Here are crystal structures of nolitinib. This is the part that reaches the adenine binding pocket. And this is the methylphenyl, still similar to imatinib, where the nitrogen in here forms interactions with the threonine 315, and this part of the drug reaches the allosteric site. لاحظوا the size of the trifluoromethyl. Okay, remember حكيت لكم behind 4-APL in particular, there is a binding pocket that is adjacent أو مكملة للallosteric site where this trifluorophenyl as the trifluoromethyl can reach into. So they've exploited that pocket of that cavity and they've inserted the trifluoromethyl حتى إنها ترتبط فيها ويصير عندي البايندينج affinity كثير أعلى. Okay, look at the part that is highlighted in yellow. What does it represent? Yeah, it's the DFG motif. Okay, this is the phenyl. This is the aspartic acid. ولاحظوا كيف بتكون عندي aspartic acid as if it's pointing out or away from the ATP binding site. The dasatinib, as we've mentioned, if you look at its crystal structure, it's actually the thiazole ring, or the nitrogen of the thiazole is the one that is forming interactions with the adenine binding pocket. Okay, so it's a type 1 as the crystal structure shows. هلا حكينا احنا يعني أنا سألتكم before إنه what do you think or what would be your approaches or strategies to counteract the resistance mechanisms اللي موجودة عندي في الcancerous cells. وإن particular نربطها بالإيماتينب. أول approach حكيته عنه إنه to design drugs that do not rely on the threonine interaction. Okay, well, that's actually one of the approaches that they've been working on, giving rise to the development of tozasertib. Tozasertib is a kinase inhibitor of APL that does not rely on the interaction of a 3-threonine 315. Okay, it forms interaction with other amino acids mostly بتكون عندي أو some of them are conserved in the APL active site. Okay, مش راح تنسألوا في الامتحان أو في الامتحان ما راح أسألكم عن التوزاسيرتيف structure ولا هو type 1 ولا type 2 لأنه البايندينج مود لإله بداخل الactive site of the APL kinase يختلف تماما عن type 1 و type 2. تكون عندي هاي الرينج كلها وكأنها أو in the front of the adenine binding pocket. البايندينج مود تماما بيختلف فما راح نطبق عليها انها type 1 ولا type 2. Okay, just understand يعني for those let's say strategies just understand the overall concept of them. الexamples اللي واردة فيهم ما تحفظوهم. Okay, to the certain structure, how it binds, the interactions اللي بيعملها as shown in this map. You don't have to worry about those at all. Okay. هلا another strategy إنه إحنا نصير نتدخل بالinteractions اللي بتصير ما بين البروتين والكاينيز. يعني recall إنه كل الكاينيز inhibitors اللي إحنا عم نناقش فيهم are those which do enter the active site of the ATP or where ATP binds in. Okay. But don't forget, those kinases interact with substrate proteins from the outside or on the surface. So many approaches now, مش بس هاي لا تنطبق على ال APL kinase, many, let's say, pharmaceutical companies بيشتغلوا على development of drugs that can insert themselves between the proteins وبيمنعوا الانتراكشن ما بين the proteins. Okay? An example for that is this investigational drug that is still, let's say, under clinical trials. ON with this number stating, let's say, this compound, which as I have mentioned, this is still investigational. This drug is being studied or was designed to interact to interact with the kinase and the protein that the kinase binds to where they at the interface بحيث انه يمنع الانتراكشن ما بيناتهم بيمنع الكاينيز من انه يعمل phosphorylation to that target substrate. Okay. Another approach واللي already احنا had an example for اللي هي development of inhibitors that have dual أو multi specificity أو بمعنى آخر إنهم يرتبطوا ب more than one kinase اللي already known إنهم to be implicated in cancer such as مثلا باللوكيميا اللي حكينا عنها where ال APL أو ال PCR APL and also ال SRC both enzymes are known to be overly active and upregulated in those cancers. So if you have one drug that can bind both and can inhibit both, let's say this would have as if you are having a synergistic effect. Okay, example عليها اللي حكينا عنه اللي هو الداساتينب. Another drug that is known to bind to both اللي هو البوسوتينب. Type 1 ولا type 2? Type 1, yep. That's the part that would bind to the, yes. And this is would reach the hydrophobic pocket and for sure this piperazine ring would go to the solvent region. Okay? The crystal structure of this drug. Another approach اللي هي development of allosteric inhibitors أو inhibitors that do not reach even the ATP binding site أو the pockets that are around the ATP binding site. لما نحكي allosteric, yes, a completely different binding site, such as for instance GNF2. That's an allosteric inhibitor or an allosteric modulator of this enzyme, which is highlighted in red. لاحظوا وين عندي بيرتبط حتى في الC loop that is way away from the ATP binding site. Another even more successful approach would be given a combination therapy of allosteric inhibitors and ATP, let's say, inhibitors. طيب آخر شيء بدنا نحكي عنه regarding the kinase inhibitors, something we've already talked about. صح احنا بنحكي selectivity ضرورية in designing any drug, لكن for kinases in particular, لأنه أحيانا in many cancers you can see more than one kinase being overly expressed أو upregulated أو overly active. designing drugs that can bind to those kinases in particular أو selective ل more than one kinase or having multi-effect على more than one kinase, this would be as if you are combining more than one drug together that are acting synergistically. Okay? طبعا هو صح a very great approach, لكن somehow when it comes to designing such drugs it's a hard task. يعني صعب جدا إنه تصمم drug بيكون بيرتبط مثلا ب more than one kinase ويكون selective لأنهم ما بيرتبط ب what we call off-target kinases. لكن still there are a few kinase inhibitors اللي إحنا بنعتبرهم أو يعتبروا let's say multi-tyrosine kinase inhibitors that are FDA approved, such as for instance sorafenib and sunitinib and so on. These are mostly involved in inhibiting kinases that are included or implicated in angiogenesis. Okay? رح نحكي فقط عن the development of sorafenib, that would be it. Okay? هلا recall إنه حكينا يعني one of the approaches via which we can design and discover kinase inhibitors إنه بنعمل إحدى screening. يعني مثلا لما أكون عارف إنه في عندي a kinase معين معروف إنه upregulated in a cancer. One way or one approach that is somehow conventional in pharmaceutical companies إنه كل ال compounds اللي موجودة عندهم في اللايبرري بصيروا يعملوا screening عليها. For sorafenib development in particular, that company that developed this drug كان عندهم يعني what they've done, they've screened تقريبا 200,000 compounds موجودين in their library against that kinase until they reached or they found out this lead compound that had an IC50 of 17 micromolar واللي تعادل كم النانومولر؟ لا Yeah, 17,000 nanomolar. Okay, واحنا دائما بال IC50 or when looking for potent inhibitors, we aim for single digit or two digit maximum nanomolar. Okay, this is a value that we can consider, and this is a potent, strong inhibitor. Okay, so بعتبره عشان هيك it's definitely not a drug candidate, لكن for sure it's a good start or a good خلينا نحكي a lead compound from which I can start with. هلا conventionally واللي احنا يعني بهيك بمختبراتنا المتواضعه اللي بنعمله انه if I have a lead compound بصير اعمل single let's say modifications at a time. All right, that's a conventional method where for instance what they've done for this lead compound, they've substituted that hydrogen with a methyl at one time, okay? And they've substituted that hydrogen with a phenoxy at another time, okay? هلا when they've substituted the hydrogen with a methyl, it gave rise to enhancement or decrease in activity. لأنه كل ما قلت عندي ال IC50 كل ما كان أفضل، تقريبا 10 folds increase بالactivity. لكن when they've substituted it with a phenoxy, الاكتيفيتي صارت عندي جدا سيئة. يعني تقريبا لما نحكي poor activity احنا بنحكي عن an IC50 of 50 micromolar and more. Okay? هلا generally أو conventionally if I have أو يعني if I have such results الفينوكسي بشيلها تماما من بالي. Any future analogs ممكن أفكر فيها بلغي الفينوكسي منها. Okay? That's conventionally. لكن a new approach for analog design, especially طبعا هاي مطبقة بالpharmaceutical companies because they have resources يصنعوا عدد هائل من let's say الانالوجز اللي هي parallel synthesis. okay? إنه أنا بصير أغير yes إنه more than one substitution and combine them together. تمام؟ فطبعا why let's say this is a very successful approach لأنه الفينوكسي for instance if combined with this ring واللي احنا بنسميها ايش؟ This is sulfur. Thiophene, okay? It gave rise to bad activity. لكن if combined هاي واحدة من الانالوجز اللي صنعوها if combined that phenoxy group with an isoxazole على سبيل المثال look at the IC50. It became even way better than a lead compound. Okay? So this is the point of what we call or consider parallel synthesis. إنه أحيانا one modification ممكن يعطيني bad activity, but if you make more than one change at the same time, this may give rise to even way more enhanced activity أو خلينا نحكي يعني much better potency towards that target kinase. Okay, this is what I just need you to understand from this slide اللي هي ال variations أو what is meant by parallel synthesis. إنه أنا أصنع أو when designing analogs إنه ما أعتمد بس على one single modifications وهي اللي تعطيني أو هي اللي من خلالها أحكم إنه where to go forward. ال parallel synthesis إنه أغير كل شيء almost at the same time. تقريبا هاي الشركة صنعت more than 1000 analog based على ال parallel synthesis. يعني they thought about every possible different combination between the different compartments of the lead compound. هلا لما تحكي مثلا 1000 analog لا بتكون every possible thing سواء مثلا الان سيليكو studies او الان سيليكو مثلا docking اعطاني انه there is good binding ولا لا they made it. Yes. And أكيد يعني you don't have to know exactly إيش ال other analogs اللي صنعوهم لكن eventually they've got to السورافينب which is the خلينا نحكي the final let's say analog that had an IC50 of 12 nanomolar. That's a very potent kinase inhibitor. Is it type 1 or type 2? شو رأيكم؟ شو رأيكم؟ بالعكس واضح نوعا ما. Which ring you think would form a binding with the hinge region? Which one? Yeah, this one that has the nitrogen is called pyridine. This one? لا usually بال let's say because it can يعني usually في الأدينين binding pocket look for rings that are planar. هلا لما تحكي عن let's say the urea group مع هاي two let's say rings that are attached to it they can flop or they can let's say rotate بحيث انهم they may clash with the amino acids up and down the adenine binding pocket. ف between اليوريا والبيريدين I would choose البيريدين. Okay, so that's the part that will bind the adenine pocket, and for sure whatever is linked to it can reach to the hydrophobic pocket and the rest of the structure to the allosteric site. So it's actually a type 2. Sorafenib is a type 2 inhibitor. You mean this is where at the hinge region, high hydrophobic, allosteric? هلا إذا كانت هاي الهيدروفوبيك وهاي الهنج ريجن هاي بالهنج يفترض الالوستريك تكون مرتبطة هون. because they need to be constitutively. هلا لو كان type 1 I would let's say break the bond in here أو حتى هون. Yes. Okay. هلا exactly the binding of those two drugs inside I'm not sure about يعني يعني there is definitely no crystal structures لأنهم weak binders فصعب جدا إنك تعملي crystallization for such a complex to begin with. My guess would be the difference between الثايوفين والisoxazole إنه ممكن يعني this group in here طبعا هي مش بس الثايوفين و this group that is attached to it. This group to fit in يعني لو افترضنا إنه it inserts itself inside a certain pocket بالactive site, okay, it may affect the relative position of the phenoxy. فممكن تكون البوزيشن لإلها تضطر إنها تتحرك بحيث إنه يصير في بينها وبين wherever أو where it sits بيكون في عندي clashes. But في حالة لما يكون عندي الايزوكسازول and there's nothing attached to it, okay, معناها بيكون في عندي some relief ما يعني يعني somehow بيكون عندي الفينوكسي able to move at a point أو in a relative position بحيث إنها تكون fits exactly inside where it needs to be binding. يعني this is my خلينا نحكي هيك humble explanation for that. يعني صح هاي يعني كل هاي rings somehow attached to each other. بالتالي their relative conformation أو where they anchor themselves at one point رح يأثر على ال other ring systems. فأحيانا just one simple, let's say, rotation or one simple change بالconformation ممكن يخلي a drug that binds well in the active site وأحيانا لا. This could be an explanation for that. ممكن يكون سبب آخر يعني بس for that يعني you need to dock those two ligands inside the active site وتشوف ال binding energy هل في clashes ولا لا، ال overall conformation كيف بتغير أو بتأثر لما أنت تغيري مثلا الثايوفين with an isoxazole. بس أنا برأيي هي it doesn't depend exactly على أنه غيرت الثيازول into an isoxazole. It's actually also that substituent على الثيازول itself. له دور كبير in my opinion. Okay. Sorafenib, yes, yes. It has, let's say, more than just one target. Yep. أكيد the binding affinity between one and another is somehow there are variations, but still it is considered somehow potent inhibitors of more than one tyrosine kinase. Any other question? طيب. سوري إنه أنا نسيت أذكرها. طيب هلا هي ذكرتني ب something we mentioned بالإيماتينب development, which is this مثل خلينا نرجع للكريستال structure بتكون يعني أوضح. هلا what is meant by that طبعا أو that مثل group they call it a conformational blocker مش لأنه it limits يعني it limits at some point let's say the number of conformations of this drug لأنه لما يكون عندي مثل rotation around this bond لما يكون في يعني بيكون كثير أصعب لما أنا أحط مثل هون. Okay, but the reason why they called it a conformational blocker because that methyl group or this size of the group would make الإيماتينب somehow selective لل-APL. Okay, because of the threonine being a small group. فهلا any other kinases that have gatekeeper residues that are bigger than threonine زي مثلا الميثايونين على سبيل المثال لو كانت فينل لحالها بتفوت لو كانت مثل وعليها أو فينل with a methyl such as an imatinib it will not enter that hydrophobic pocket. What is meant by this methyl being a conformational blocker is that it blocks the entrance of the methyl phenyl in binding pockets أو in hydrophobic pockets in other kinases اللي فيهم ال gatekeeper residue bigger than a threonine. So it aids, it's somehow a selectivity or a group that adds to the selectivity of imatinib. Okay. Any other question? طيب. يعطيكم ألف عافية. هلا مبدئيا الامتحان رح يكون بالانتي نيوبلاستكس والكاينيز انهبيتورز رح نضيفها لمادة ليكويز. تمام؟